Hello, Sanyalan, Dumanan, Maureen, Abhishek, Nda. I'm Miss Ndango. I'm with my colleagues, Miss Nisi, Miss Chawoge, and Mr. Zulu, who are from that School of Education. So in grade 12, today we are going to talk about DNA, the code of life. So we'll try by all means to break down the word DNA. DNA is just the word that is abbreviated into 3 letters, which D stand for deoxyribose, which is the sugar that has lost oxygen from the second position of the carbon atom. And then N stands for nuclear, which represents the nucleotide that could be any of the 4 standard nucleotides. It could be adenine, thymine, guanine, cytosine. But you guys going to go through that part with Miss Nisi. So let's just define DNA. DNA is the molecule that carries the genetic material for the development and functioning of an organism, where genetic information acts in the genetic test of the person and the family members. So grade 12, I'm going to hand over to my colleague, which is Miss Nisi, to go through with you the second part.
As already my colleague have already introduced me, today we'll be doing the code of life, which is in DNA, which is the new topic that the grade 12 will be doing. As we know that in grade 11 we did organic compounds of carbohydrates, proteins, lipids, and vitamins and nucleic acids. So we're gonna do a quick recap on them and what are their monomers. Carbohydrates, their monomers is monosaccharides. Proteins, their monomers are amino acids. Lipids, their building blocks is fatty acid and glycerol. Vitamins, there are no monomers for vitamins and minerals. Nucleic acids, their building block is nucleotide.
Hello everyone. As introduced, my name is Mr. Zulu, and in this part of the video I shall be talking about DNA composition. As we all know that DNA is a molecule that is a polymer of nucleotides with 2 linked strands that wind around each other, forming a shape of a double helix. Now we know a helix shape is something that looks like 2 intertwined objects, or say molecules. Now each nucleotide is composed of a nitrogenous base, a 5-carbon sugar, which in this context of a DNA will be a deoxyribose sugar, and also a phosphate group. Now there are 4 nitrogenous bases that are contained in a DNA molecule, and 2 of these nitrogenous bases they belong under purines, which are the adenine nitrogenous base and guanine nitrogenous base. And then the other 2 they fall under pyrimidines, which are the cytosine nitrogenous base and the thymine nitrogenous base. So in a nutshell, this diagram here represents a situation of the composition of a DNA molecule. So what you will have here is your phosphate group, and then what you'll have here is the sugar, which in this context would be a deoxyribose sugar, and then here will be your nitrogenous base that will contain any of the 4 nitrogenous bases, which would be adenine, thymine, cytosine, or guanine.
I'm going to do a quick recap on what my colleagues have already did. We have 2 types of nucleic acid, DNA and RNA, deoxyribonucleic acid and ribonucleic acid. Both types are made of smaller units, building blocks or monomers called nucleotides. Each nucleotide is made up of the following: a nitrogenous base, a sugar portion. In DNA, sugar is deoxyribose, and RNA, sugar is ribose. A phosphate portion. Next, I'm going to show you an example of a nucleotide that I was talking about. I know that my colleague have already shown you this. I'm just going to explain further. We have a phosphate portion, we have sugar portion, and nitrogenous base. Phosphate portion is always attached to the sugar portion. Nitrogenous base is always attached to sugar portion. In sugar portion, we have deoxyribose and ribose. It depends on what we are talking about. Since we are talking about DNA, then in sugar portion, we're going to have deoxyribose. Phosphate group will never be attached to nitrogenous base. And on the next slides, we're going to talk about where we find DNA and the functions of DNA. As our today's lesson is going to be about DNA, deoxyribonucleic acid, there are three places in which DNA is found. Number one, we have nucleic DNA, which we call it an extranucleic DNA. And we have mitochondrial DNA, which partially is important in tracing our ancestors. We have chloroplastic DNA. We have functions of DNA. Nucleic DNA controls the synthesis or manufacturing of proteins. DNA controls the synthesis of enzymes or enzyme specialized proteins that regulate and control metabolic activities. Nucleic DNA transmit heredity from parents to offsprings, which this means information from existing cells to new produced cells.
I am going to be continuing with the discovery of DNA. DNA was first discovered in 1969 by a Swiss biochemist Friedrich, where he discovered that there is a substance in the nucleic acid. In the early 20s, scientists studied that and suggested that a nucleic acid played a crucial role in the genetic transmission and then heredity. In 1944, Oswin Arberty, Collins, and McClary conducted an experiment that proved that DNA and not protein was responsible for transmitting genetic information. They showed that DNA was extracted from a bacteria and then transferred to another one. After the transmission, the organism that received the DNA from the bacteria, it actually grew more hereditary characteristics that are from the bacteria that show that DNA is the one that carries our genes and hereditary information. In the early 50s, Rosalind Franklin used an X-ray to show and create an image of what a DNA is, whereby there was a DNA fiber, and he worked on a double helix structure. Here is an example of a double helix structure of what Rosalind discovered from his X-ray. Going forward, we are going to this side. We are going to this side to be looking at what James and Watson Crick did on top of Rosalind's work, whereby they discovered that the helix structure described by Rosalind has actually characteristics that it is able to replicate, and then they actually showed that there's also micronucleic acids, that is adenine, ethyl, cytosine, and guanine. As I was saying, Francis and Watson actually discovered that, can I move the camera to the side? Actually discovered that the DNA can actually replicate and make two of its same structure. It actually replicates but does not change the genetic information. They also discovered that the double helix actually has components inside, which is the adenine and the adenine and thymine that are what? Paired together by a bond. And then there's also a cytosine and a guanine that are also held together by a bond. These four components actually repeat itself until the gene is completed, which is the what? It can also be A, T, C, G in a repeating pattern. That is all about discovery of DNA. Thank you.
Deoxyribonucleic acid contains your genetic code, DNA, a molecule that makes up all your chromosomes. Double helix structure, a two-stranded spiral. Set of instructions for your cells so that they know what to do, like send their signals and make new bonds. Making proteins from acid to amino. Complete set of genetic instructions is a genome. Nucleotides like genes and half the things that they're composed of phosphate, nitrogen, base, and deoxyribose. These nucleotide bases made up from these small attending bonding diamonds, adenosine, and that's all. A always pair with D, and that's the base barrel. C always pair with G, and that's the way they go. Deoxyribonucleic. DNA is deoxyribonucleic acid, yeah. Deoxyribonucleic. DNA is deoxyribonucleic acid. Complementary bases, each on opposite strands. You can't remember the pairs, both a mnemonic GCAT. Control complex process to make a protein from a gene. It goes transcription and translation to intercepts in the journey. Transcription takes information, transfers to mRNA. Messenger moves the nucleus and onto the next stage. This is called translation. Happens in the cytoplasm, ribosomes read the